cellular respiration, or how we make ATP from different macromolecules, so from food. How are we going to make ATP energy from food? How does that work? So organisms must take in energy from the outside to be able to survive, to continue on living. And energy is derived from organic molecules like glucose or lipids. Any of the macromolecules we talked about in the biochem unit are where we're going to get those molecules as heterotrophs. Things like plants make those molecules via photosynthesis using the sun's energy. Glucose in specific is broken down in cellular respiration to form ATP. So we're going from glucose to ATP. So all those other macromolecules we can break down into some other parts that will also feature in or can be used in this cycle, but we're going to focus on glucose because a lot of macromolecules like carbohydrates can be broken into glucose or glucose residues. So if we think about what we're going to focusing on, cellular respiration, that happens in the mitochondria. We take organic molecules and oxygen and turn them into ATP energy for the cell to use. So energy in food is stored in as carbohydrates, proteins, and fats, so our macros. Before that energy can be used by the cell, we have to convert it to ATP through cell respiration. So cellular respiration converts the energy stored in these molecules into ATP, or adenosine triphosphate, like we talked about last time. So the basics that we need to know is aerobic respiration. So in the presence of oxygen, releases energy by breaking down food in the presence of oxygen. So we have glucose plus oxygen equals carbon dioxide plus water plus 36 molecules of ATP energy. There's another form of respiration called fermentation, and that is when we only partially break down glucose without oxygen. A lot of bacteria does this when you ferment alcohol or ferment things into alcohol. This is what's happening. It releases only a small amount of ATP, so it's not as efficient as aerobic respiration, which we'll focus on today. Glycolysis specifically is the first step of cellular respiration. We basically split a glucose molecule into two pyruvate molecules. One glucose molecule into one 6-carbon glucose molecule into two 3-carbon pyruvate molecules to make, to start harvesting energy. So we start harvesting energy using that breakdown. So the energy that we use eventually to make ATP is stored in electrons. So the transfer of electrons during chemical reactions releases energy stored in that, and we're going to use that to make ATP. So we are going to use electrons to make ATP. Now, electrons are going to be our energy stores, our batteries. Oxidation-reduction reactions are those things that involve the transfer of electrons from one substance to another. So basically, cellular respiration deals with a lot of oxidation-reduction reactions to harvest those electrons as we digest glucose to get the energy out of them to use to make ATP. So chemical reactions that transfer electrons are called redox reaction. In oxidation, one substance loses electrons, and in reduction, one substance gains electrons. In cellular respiration, glucose and those substances it gets broken down into, are continually oxidized to harvest those electrons by an oxidizing agent, by a chemical that gets reduced and stores those electrons to use later to transfer into ATP. So, our basic cellular respiration can be viewed as a redox reaction. Glucose becomes oxidized. We take those electrons from, as we break down glucose, and we use them to make energy. We make energy by gradually giving those electrons to oxygen that we breathe in, by reducing oxygen. So if we take the electrons from glucose and we give them to oxygen to form water, we get energy out. So A is oxidized, glucose is oxidized. We store the electrons in a transfer molecule before they go to oxygen, and in that process make energy. NAD+, or nicotine amide adenine dinucleotide. Nicotinamide adenine dinucleotide is an electron carrier. So when glucose is broken down into a number of steps, as it's broken down, the electrons from glucose are transferred to nicotinamide adenine dinucleotide as a storehouse, as a storage for electrons. So NAD+ gets reduced, it gains those electrons, and it stores those electrons to convert into ATP energy. And when it is reduced, it is converted to the form of NADH. So this means energy potential. This means we have some electrons. And this is what NAD+ and NADH look like. Alright. Those electrons stored in NADH pass through an electron transport chain, which is a series of proteins in the inner mitochondria where work can be done with them. So those electrons end up ultimately making ATP in our electron transport chain. And this won't be the focus of this section, but just like in your book, this is kind of a picture of what that third step we'll look at next time goes over. So in cellular respiration, there's 3 stages. 1, glycolysis, for breaking down glucose into 2 molecules of pyruvate, getting some energy, making some NADH, but mainly getting it ready for later steps. 2, the citric acid cycle. This completes the breakdown of glucose. We get more NADH, some ATP out of there. And 3, the electron transport chain. Talk about that next time. That's where we get lots of ATP energy. Use all those electrons to make ATP energy. So the overview. We're going to go through glycolysis first. Glucose gets converted to pyruvate. We have some ATP that comes out through substrate level phosphorylation. Then we go on to the citric acid cycle. Some more ATP comes out through substrate level phosphorylation, but mainly we get a lot of NADH and FADH, which are both store electrons. And electrons, remember, are those potential energy. And then later we will talk about the electron transport chain, where we get out a lot of ATP through oxidative phosphorylation. So phosphorylation is adding a phosphate, to ADP to make ATP using an enzyme. So substrate level phosphorylation, we're sticking this molecule of ATP, of phosphorus, onto ADP to make ATP using an enzyme in a specific step. Not much ATP is created this way. Oxidative phosphorylation is phosphate is added to ADP to make ATP by ATP synthase, a protein-embedded pump in the mitochondria. Lots of ATP are made through this step. Glycolysis and the citric acid or Kreb cycle only makes ATP this way. That's why it can't make a lot. We'll show you. But they do make a lot of NAD+ and FAD. So, step 1, glycolysis. In the first series of reactions, glucose is split into 2 molecules of pyruvic acid, C6 broken to C3. This occurs in the cytoplasm of the cells and does not require oxygen. So this is the only step that something that is going through fermentation can do. It releases only 2 ATP molecules, not enough for most living organisms. Glycolysis' main step is to prepare glucose into pyruvate so that it can be used to harvest a lot of NADH in the citric acid cycle, and it produces some NADH by itself. So they're kind of 2 halves, and we don't need to memorize any of these names or these enzymes, but we want to see that what happens is at the beginning, in 2 steps, we're sticking phosphate groups on to our glucose molecule, and that costs 2 ATP. We're preparing this glucose molecule to be used later, specifically in the citric acid cycle. And in the second step of glycolysis, we're getting some energy from that glucose molecule by breaking it down to pyruvate. So we get 2 ATP out here and 2 ATP out here by sticking these phosphates onto ADP. We also make 2 NADH, which are electron carriers for electron transport chain. We're storing up electric energy, electrons potential in the form of NADH for the electron transport chain later. So, in all, in glycolysis, we start out with glucose. At the end, we end up with 2 molecules of pyruvate. It takes 2 ATP to invest to break it down, and we get 4 ATP out and 2 NADH out. So in total, we gain 2 ATP and 2 NADH. So some ATP directly, and then 2 molecules of NADH, which can be later turned into ATP. Next, the citric acid cycle. The citric acid cycle, also called the Kreb cycle, completes the breakdown of pyruvate and releases the energy from glucose in the form of NADH and ATP. This occurs in the matrix, so the inner part of the mitochondria. In the presence of oxygen, pyruvate enters the mitochondria. Before the pyruvate can enter the citric acid cycle, it must be converted to Acetyl Co-A, and this is in a process called pyruvate oxidation. Getting some NADH out by further preparing pyruvate to be converted to harness the energy, to take those electrons to oxidize it. So we're taking pyruvate, creating Acetyl Co-A. Some energy, like we said, released in NADH. The Acetyl Co-A is what goes into the citric acid cycle. And here's a picture from your book of pyruvate being converted to Acetyl Co-A and releasing more energy, NADH. The Acetyl Co-A then enters the citric acid cycle in the matrix of the mitochondria. The citric acid cycle breaks down Acetyl Co-A in a series of steps that releases carbon dioxide. This is where the carbon dioxide you exhale comes from. This citric acid cycle produces 1 ATP, 3 NADH, so electron carriers storing energy for the electron transport chain, and 1 FADH2. That's just like NADH, that stores electrons per turn. So it goes once around, 1 turn per Acetyl Co-A, and we get 2 Acetyl Co-A's from each glucose molecule. So 2 turns per glucose molecule. And you can see here what's produced. 2 carbon dioxides, 2 ATP energies, and then all of these NADHs. We get 6 NADHs in total for 1 glucose molecule, 2 FADHs, and then 2 NADHs from our pyruvate digestion, so oxidation. So all this potential electrons to use an electron transport chain, 2 ATP from one glucose molecule, so 2 turns. It harvests this energy by 8 steps in the citric acid cycle. You don't have to remember these, but you need to remember that we get out this NADH and this ATP through substrate level phosphorylation, these NADHs and this FADH2 through oxidation. So we continually oxidize to remove those electrons to use for later. So the key in this whole thing is we want to gather electrons for the electron transport chain. That's where we get lots of ATP from. In the process of doing that, for one glucose molecule, we get from glycolysis, we get ATP and NADH, and from the citric acid cycle, we also get ATP and NADH. Alright, this is just the background. We're going to go through this a lot more detail on Tuesday. But this just to get started. Remember, keep it simple. Don't remember all of these names and things like that. What's coming in, what do we get out, and where is the energy going? Usually stored in NADH or FADH, and sometimes converted directly to ATP through substrate level phosphorylation.